In this video, we will begin by reviewing genes and their expression. We will then learn how environmental factors can affect gene expression and examine the relationships between light intensity, chlorophyll synthesis, and the rate of photosynthesis in plants. Let's begin by defining some of the key terms we will be using throughout the video. A gene is a section of DNA that contains the information needed to produce a certain characteristic or functional unit. For example, proteins are a type of functional unit. And many genes contain the information needed to produce proteins like enzymes and hormones. Gene expression is the process by which the information encoded in a gene is used to create or synthesize proteins. Due to the information they carry, genes are partly responsible for how an organism looks and behaves. For example, genes code for the proteins that determine eye color, which can vary considerably among individuals. And genes can influence a person's tendency to be more shy or more social. However, an organism's environment can cause the level of gene expression to vary. And these variations in gene expression can in turn affect an organism's appearance or its behavior. A rather extreme example of the influence of environment on gene expression is seen in sea turtles. The temperature experienced by sea turtle eggs determines the sex of the embryo inside. This in turn determines the sex ratio of the hatchlings. When the temperatures experienced by the eggs are warmer, the hatchlings will be primarily female. When temperatures are colder, the hatchlings will be primarily male. And intermediate temperatures lead to a sex ratio of approximately 50-50. This phenomenon occurs because the temperature experienced during egg incubation changes the expression of genes that code for various enzymes and sex hormones. Scientists continue to research the evolutionary advantage of this temperature-dependent sex determination. In plants, environmental factors such as carbon dioxide concentration, temperature, and light intensity affect the expression of genes involved in photosynthesis. You probably already know that photosynthesis is the process by which plants use light, carbon dioxide, and water to produce glucose and oxygen. You may also remember that photosynthesis takes place inside plant organelles called chloroplasts. If we zoom in to get a closer look at a chloroplast, we can see a structure inside called the thylakoid membrane. And embedded in the thylakoid membrane is the molecule chlorophyll. Chlorophyll is a biological pigment, which is a molecule that absorbs specific wavelengths of light while reflecting other wavelengths. Chlorophyll absorbs blue and red wavelengths of light more efficiently than green wavelengths. This means more green light is available to be reflected by the plant, giving them their characteristic green appearance. Inside the chloroplasts, chlorophyll is constantly being broken down. So the plant must continually synthesize new chlorophyll in order to maintain photosynthesis. In fact, the amount of available chlorophyll is one of the main factors affecting the rate of photosynthesis. And the environmental factors that affect gene expression influence the amount of available chlorophyll. Now let's take a closer look at how the environmental factor of light intensity interacts with available chlorophyll to affect the rate of photosynthesis. This simplified graph has light intensity on the x-axis and photosynthetic rate on the y-axis. Starting at point A and moving right along the x-axis, we can see that as light intensity increases, photosynthetic rate also increases. This relationship exists because light intensity influences chlorophyll synthesis in plants. Under low light conditions, the genes involved in chlorophyll synthesis, shown here as blue bars on a strand of DNA, will be downregulated. This means the plant will produce fewer of the proteins needed to make chlorophyll, and the rate of photosynthesis will be low. If the plant remains in these low light conditions, it may become yellow. In some cases, the plant may die, but this process also occurs naturally, such as when trees lose their leaves in the fall. This scenario of low light and low photosynthetic rate is represented by point A on the graph. Under conditions of high light, the genes involved in chlorophyll synthesis will be upregulated. The plant will produce plenty of the proteins needed to make chlorophyll, and the rate of photosynthesis will be high. Plants that are grown under conditions of high light intensity are typically greener and healthier than plants grown in conditions of low light. This scenario of high light intensity and high photosynthetic rate is represented by point B on the graph. Increasing light intensity cannot increase photosynthetic rate indefinitely. And we can see this on the graph. After point B, the curve levels off or plateaus, and the rate of photosynthesis becomes stable. This occurs because other factors become limiting, so further increases in chlorophyll synthesis alone won't allow the plant to increase its photosynthetic rate. However, changing another environmental factor, such as increasing the temperature to a more optimal level, will allow the plant to increase its photosynthetic rate relative to the colder temperature. But even in this scenario, the curve eventually plateaus and photosynthetic rate remains stable, because neither light intensity nor temperature are limiting factors. Now that we've seen how light intensity affects chlorophyll synthesis through gene expression, which in turn affects photosynthetic rate, let's try a practice question. Chlorophyll is required by the chloroplasts of leaves to capture light. For what process do the chloroplasts require this light energy? You may remember that chlorophyll is the green pigment found within the thylakoid membrane of chloroplasts. A pigment is a molecule that absorbs specific wavelengths of light and reflects other wavelengths. In the case of chlorophyll, blue and red wavelengths of light are absorbed more efficiently than green wavelengths of light. Inside the chloroplasts, this absorbed light energy is used to break apart water molecules, releasing protons and electrons that are used to create chemical energy. The chemical energy is then used to fix the carbon from carbon dioxide into glucose and to release oxygen as a product. The name for this process is photosynthesis, and it's how plants make their own nutrition to survive and grow. Therefore, the name of the process that chloroplasts require to capture light energy is photosynthesis. Now let's try a final practice question. 3 groups of plants belonging to the same species are exposed to different intensities of light. Group 1 was exposed to the light intensity shown for 12 hours, Group 2 for 24 hours, and Group 3 for 36 hours. The graph provided shows the amount of chlorophyll synthesized by these groups of plants at different light intensities. What can be concluded from this graph? A. For all light intensities, more chlorophyll is synthesized when the plant is exposed to the light for 12 hours than when it is exposed to the light for 36 or 24 hours. B. For all light intensities, more chlorophyll is synthesized when the plant is exposed to the light for 24 hours than when it is exposed to the light for 36 or 12 hours. C. The intensity of light does not have an effect on how much chlorophyll is synthesized by a plant, but the duration of exposure does. D. For all light intensities, more chlorophyll is synthesized when the plant is exposed to the light for 36 hours than when it is exposed to the light for 24 or 12 hours. The graph shows light intensity in lux on the x-axis and the amount of chlorophyll being synthesized by the plant in milligrams per gram on the y-axis. The 3 curves represent the 3 groups of plants that were exposed to different light intensities. Group 1 for 12 hours, Group 2 for 24 hours, and Group 3 for 36 hours. We can see that all 3 groups had an increase in chlorophyll synthesis as light intensity increased from 0 to 5,000 lux. And from this we can make a conclusion that an initial increase in light intensity also increases chlorophyll synthesis. We can also see that at 0 lux, Group 3, the 36-hour group, synthesized more chlorophyll than Groups 1 or 2. And this pattern holds true for every light intensity. So, a second conclusion we can make is that Group 3 synthesized the most chlorophyll at every light intensity. We now have enough information to answer the question. So let's bring back the answer options one at a time to determine the correct one. For all light intensities, more chlorophyll is synthesized when the plant is exposed to the light for 12 hours than when it is exposed to the light for 36 or 24 hours. We concluded that group 3, the 36-hour group, synthesized the most chlorophyll at all light intensities. So we can rule this answer choice out. For all light intensities, more chlorophyll is synthesized when the plant is exposed to the light for 24 hours than when it is exposed to the light for 36 or 12 hours. As with the previous answer, we can rule this one out because we know that group 3, the 36-hour group, synthesized the most chlorophyll at all light intensities. The intensity of light does not have an effect on how much chlorophyll is synthesized by a plant, but the duration of exposure does. While our first conclusion was that an increase in light intensity initially increases chlorophyll synthesis, so we can rule this answer choice out. For all light intensities, more chlorophyll is synthesized when the plant is exposed to the light for 36 hours than when it is exposed to the light for 24 or 12 hours. This answer choice is correct because it restates our second conclusion about group 3. Now let's review some of the key points from the video. Gene expression leads to the synthesis of proteins. The level of gene expression can vary, and this variation is influenced by an organism's environment. Chloroplasts are the site of photosynthesis, and they contain the green pigment chlorophyll. In plants, exposure to light affects the expression of genes involved in chlorophyll synthesis. If light is a limiting factor, a higher light intensity will increase chlorophyll synthesis, while lower light intensities decrease chlorophyll synthesis. In turn, an increase in chlorophyll synthesis leads to a higher photosynthetic rate, while a decrease in chlorophyll synthesis leads to a lower photosynthetic rate. When light is no longer a limiting factor, the rate of photosynthesis plateaus.